Hi, I'm Jay Phelan. Let's talk about some biology. We're going to talk today about a type of extension to Mendelian genetic thinking that we call suppression. And it is, in essence, a way of looking at some of the complexity when we are going from genotypes to phenotypes. So, as we know, Mendel talked about 7 different traits, but all the traits he talked about were coded for by a single gene. There were 2 alleles. One was completely dominant to the other. There were no environmental effects. And that worked for everything he looked at in the pea plants. However, we want to have a bigger theory of how genotypes are converted into phenotypes, how inheritance passes from generation to generation, because many, many traits are coded for by more than one gene. So we're going to look at another case today where 2 different genes can influence the same trait. So let's say you're working with fruit flies, Drosophila melanogaster. Here's a picture of one, and typically they have red eyes. Geneticists like to call this the wild type, as if there's sort of one standard version that flies typically have, and then anything else is a version of a mutant. We can do that or not. But in either case, most of the flies you're going to see have red eyes. Let's say, though, you're looking at them and you can work with thousands or tens of thousands of fruit flies. That's one of the advantages of using them as a model system. Let's say you see some flies with purple eyes. I've got a picture here of the purple eye phenotype, and you might think, well, I'm not sure I would call that purple, just like the other one. Do you call it red? Well, it's tricky, but people who study these have looked at lots and lots of different versions. So we see this one. Here's our standard red-eyed fruit fly, and here's one where we've got a white-eyed mutant and the wild-type red eyes. And here's a gradation of many different types of eye-color mutants that have been studied in fruit flies. So it's a very well-studied biochemical pathway involving about 20 steps, and we've looked at lots and lots of different genotypes that we have and phenotypes that we see. People have gotten pretty good at distinguishing subtle differences between the various shades and then investigating what the underlying cause of those phenotypes is. So back to our red-eyed fly, we come across some purple ones. Let's say they're new, so we're curious. Where'd that come from? Do what any geneticist would do. What's that? Cross them. So we cross these flies. We get an F1 generation of all red-eyed flies. And I should say, when I say we're going to cross them, I mean we're looking at true breeding red-eyed flies, true breeding purple-eyed flies, meaning that they are populations where breeding them with each other always gives you the same phenotype. So in this case, we cross them, and we always want to choose the most parsimonious explanation for things until we're confronted with observations that say, hey, that's not going to work anymore. You need something different or more complex. So in this case, we'd say, all right, it looks like red eyes is just dominant to purple eye. So we'll assume there's a single gene, and purple eye is a recessive mutation relative to red eyes. But we would assume here that we'd have these heterozygous flies. So let's take these F1 red-eyed flies and cross them with each other. You cross them, and think about, if it was a single gene with 2 alleles, that generation, they would all be heterozygous. And if you cross the heterozygous flies and purple is recessive, think about what proportion of the offspring would be purple, homozygous recessive. In this case, we got 13 red to every 3 purple. So there's a slight deviation from what we would have expected if a single gene with 2 alleles was coding for this. So now we got to move on to maybe not as simple an explanation, but something that fits our observations a little bit better. In this case, maybe 2 genes are influencing this trait. We say this because in the F2 generation, we got 13 red to 3 purple, and that's obviously a modification of 9 to 3 to 3 to 1, right? I know, again, it's not obvious, but I'm going to show you how you can get 13 to 3 from a cross of individuals that are heterozygous for 2 different genes. So we're going to assume now, as our next best interpretation of these data, that 2 genes are involved. I've got a cross here of double heterozygotes. So we say P, this is going to be a pigment gene. S is going to be a suppressor gene. You'll see what that means later. And the cross of the double heterozygotes, those F1s, we're assuming that from the true breeding individuals of the parental generation that we got F1s that were heterozygous for both of the genes. When you cross them, we're going to get 9 to 3 to 3 to 1 in terms of what do the genotypes look like. 9 out of 16 are going to have a dominant for at least one dominant for each of the two genes, and 3 out of 16 are going to have a dominant for one, recessive for the other. 3 out of 16 are going to have homozygous recessive for that one, and at least one dominant for the other gene. And finally, 1 out of 16 are going to be homozygous recessive for both. Now, without suppression, red's just dominant to the mutant purple allele. If you look at these, I would say, all right, yeah, 12 out of 16 are going to be red, and 4 out of 16 are going to be purple. But that's not what's going on here. We've got a situation here, and I'll work a little bit backwards just to tell you what's going on. The presence of a homozygous recessive for this second gene, the suppressor gene, what it can do is suppress the mutant effects of the pigment gene. In other words, you've got your regular pigment gene, but every now and then if there's the mutant, the little p, the suppressor can revert that back to the wild type. So what do we get here? If you've got a dominant, one copy of the dominant allele or two, that's what the straight line means. The other allele could be either of them. As long as you have one dominant, then you're going to show red. So you show the dominant trait. Doesn't matter what you have for the suppressor because you're not showing a mutant trait. If you have, in the second case, you've got homozygous recessive for the suppressor, but again, doesn't matter because you're producing the wild type pigment allele. So same thing, it's going to be red. Then we look down at the very bottom one, you are homozygous recessive for the pigment allele. In other words, you're going to show the phenotype for the mutation. You're going to show purple eyes. However, you look to the second gene. Now, little s. That is the condition in which that second gene will suppress the mutant, and it will restore the wild type. So it turns out to still be red eyes, even though if we only looked at the P gene, it should be purple eyes. Then we look at the case here where you've got little p, and big S and something else. Well, in this case, you've got little p, homozygous recessive for the mutant allele for purple eyes, and you looked at the second gene, the suppressor gene. You don't have homozygous recessive, so we can't suppress it. So the mutation shows. The mutant phenotype purple eyes shows up. 3 out of 16 are going to be purple. 13 out of 16 are going to be red eyes. And that's what we saw. So again, to restate, when you have the little s genotype, it suppresses the mutant effects of the little p genotype. And we call that suppression. We call it recessive suppression because you have to be homozygous recessive for that suppressor gene to suppress phenotypic expression of a recessive mutation at another gene. Now, that seems reasonable and relatively straightforward, but you have to ask yourself, all right, well, we're making some assumptions here that, for instance, the suppressor is only going to revert the mutant back to the wild type if it's in the homozygous recessive condition. What if it's the opposite? What if the suppressor has its suppressing effect if you've got 1 or 2 copies of the dominant allele? Could be. So we can look at that because there are cases where that's how the suppressor gene functions. It functions when you have 1 or more copies of the dominant allele. So if we did that, then what we'd find is that we have a ratio of 15 wild type to 1 mutant. And I can leave you to look at the 9 to 3 to 3 to 1 and figure out for each of those genotype classes what's the phenotype going to look at like so that you get the 15 to 1 dominant suppression. But it gets even more complex here because, all right, the suppressor can suppress when it's homozygous recessive, or it can suppress when it is heterozygous or homozygous dominant. What about the mutant pigment gene, the purple one that we said was homozygous recessive? Well, we have mutations that function as when they're recessive, homozygous recessive, but we also have mutations that show their effect in the phenotype when they're heterozygous or homozygous dominant. So opens up a whole can of worms, right? The pigment can give you the mutant in the upper left here if you've got homozygous recessive genotype. Or it can appear if you've got one or more copy of the dominant allele. The suppressor can suppress when it's homozygous recessive. That's the first case we did. Or it can suppress when it is heterozygous or homozygous dominant. That's the second case we did, the 15-1. But you'd have to also think about each of these with the case of the recessive mutant pigment allele, which is the example we did. But you'd have to redo it again if we were looking at a case where the mutant phenotype showed up for that P gene in the case of a dominant, homozygous dominant, or heterozygous case. And in the end, you're going to get these ratios. I'm not going to work them out because it's relatively straightforward. But you look at the mutant phenotype, when does it show up in the dominant or recessive condition? And then you look at the suppressor. Does it suppress that? It's kind of weird. It's like a double negative. When does it suppress the mutant, return it back to the wild type? But that's what we got in the recessive or dominant. And then you end up with this ratio of wild type to mutant. So we'll say our take-home message for this is that suppression blocks the expression of a mutant phenotype, and it restores the wild type. There are many different ways, physiological ways, biochemical ways, that this can be brought about. So I only say that it warrants an italicized parenthetical addendum to the take-home message to say that this suppressor gene might establish some new pathway that supplants some part of a pathway in the standard pathway that's messed up by the mutation. So it might be some additional pathway, and you create a compound that can now slot into this other biochemical pathway that's been blocked by a mutant. And if you produce something else, then things can continue on. That's one way that it can work. There are other ways, but the general idea of suppression, reverting mutants back to the wild type, has been very well studied in a range of organisms, and we know its effect on the phenotype ratios is as I've described. So there you have it.